Alrighty, so, all right. Hi, everybody. Welcome. And I guess maybe really welcome back to the MSA SPS, or the Mycological Society of America Student and Postdoc Section, Students Mycological Symposium. Long word or names. So what we'll be doing is today we're going to start right on the dot, right at 4 o'clock, to give our speakers enough time to talk and explain their work, and eventually have enough time for questions too. So it'll be 25 minutes per talk and then 5 minutes for questions, okay? So we have 2 speakers, so it'll fill up the hour real fast. We're excited to have you here since this is our second meeting. Our first meeting was back in January. My name is Tanya Kerbasoyan. I am a 4th year microbiology graduate student. I'm in Dr. Jason Steich's lab, and I am the vice chair of MSA SPS. Here I have Maria, who's the webmaster. I don't see Jillian here yet. She's the social media coordinator. She's the one who's sharing the posts on Facebook and Twitter. I don't see Casey yet either, but no worries. She's our treasurer. We have Rob Powers. I think I saw him earlier. He's our chair. And possibly Rebecca. No, I don't see her yet. So Rebecca's the past chair. So that makes the MSA student section. We do have some other people, but yeah, they're also not available yet. But anyways, welcome. First thing I'd like to say, if you have any questions as the presentation is going on, make sure you write them down in the chat box. And if you want, I could be the one read the question to our speakers, or I can call out your name and give you the option to, hey, if you want to ask your questions personally, you're welcome to. And again, this meeting will be recorded and is being recorded right now. So if you have privacy issues, you're welcome to have your camera off. We will be sharing the link through Facebook and Twitter and other social media aspects. And now for our 2 speakers. So they both hail from Antonis Rokas' lab at Vanderbilt University. Sorry, Jacob, I'm going to be calling you first, so you're going to be our first speaker. So Jacob Steenwyck, maybe you've seen his posts on Twitter. He posts a lot about these really cool R color palettes that he generates. I think it's such an awesome thing that he does. And so a little bit about him. He did his undergrad and master's at Clark University in Worcester, Massachusetts, and worked in the labs of John G. Gibbons and Robert Drewel. Thereafter, he joined Antonis Rokas' lab at Vanderbilt University, where he is now a Howard Hughes Medical Institute James H. Jillian pre-doctoral fellow. So together, him, Jacob, Antonis, and other collaborating labs such as Chris Hedinger at UW-Madison, Gustavo Goldman at University of Sao Paulo, and Nicholas Alberlees at UNC Greensboro. They've published over 30 manuscripts, and among them, 11, Jacob is first author, which, no pressures, right? But beyond scientific contributions, Jacob has also promoted diversity, equity, and inclusion in the sciences. For example, he's previously been the president of inclusivity in the Bioscience Association of Vanderbilt University and serves as inclusion coordinator for the evolutionary studies. During his free time, you can find him rock climbing or making art. He has made award-winning art that has been featured at multiple universities in the United States, and most recently culminated into the Sci Art Shop, which is a science-themed art shop that sells mugs, stickers, and hoodies with portraits of animals and fungi on them. But most importantly, the shop enables people to purchase with purpose because 100% of the profits go toward wildlife conservation efforts. So since opening in August 2020, he's raised over $1,000, and he's looking forward to creating more art for Earth, which is super exciting. I think you should definitely share your link in the chat so we can all peruse. And so we're gonna give Jacob the stage. You're welcome to start sharing your slides and presenting.
Yeah, thank you. Thank you so much for the introduction. Thanks for organizing and having me be a part of this. It's really exciting to be here. So yeah, despite the introduction, I kind of feel like probably most people aren't familiar with who I am. So I thought I'd take a little bit of time to do that first, and then we'll get into the science. So who I am is pretty much entirely influenced by those who raised me. And so this is a picture of my family during the pre-COVID times when my sister Emily was getting married to my now brother-in-law Josh, and then there's my other sister Nina and her husband Gary, and of course my parents Howard and Mercy. So this beautiful family is also growing, and so if you hear a baby crying, perhaps it's my sister's daughter, my niece, my goddaughter actually, and she's the newest member in the family. So as mentioned during the introduction, I do a bit of this like art science stuff on the side. And so it's culminated again into that sci art shop. Yeah, so there's stickers, mugs, other things. But the main point is that I'm trying to use art to raise awareness about about endangered species. And in a very fantastic way, I'm trying to immortalize endangered species. So the African wild dog, the blue whale, Galapagos penguin, these are all endangered species. And they're done in these Andy Warhol styling portraits to sort of connect to things people are familiar with. But of course, I cannot forget that I do love fungi. And so I have featured various fungi in the shop as well, including the very classic fly agaric, oyster mushroom, and the morel. And most recently, thanks to Gianni Liddy, he invited me to write a little opinion piece perspective in the journal Yeast, and one of the portraits of budding yeast was able to make it into the cover. So I'm really excited about that, as well as the opportunity to write about the stance I'm taking to unify or reunify rather the arts and sciences. A very Clark University liberal arts school education sort of motto. So as mentioned, it's really to raise money though, to try and save these types of organisms as well as others. And this graphic is a little outdated, but the shop has raised pretty much $1,000 at this stage. And so I'm excited to keep pushing this effort to try and, fulfill this mission of art for Earth. But of course, I'm here today to really talk about science. And so as mentioned before, all of the work that I've done to date primarily has been done with my PhD advisor, Antonis Rokas at Vanderbilt University. And he's a really wonderful advisor that has enabled me to conduct a lot of research. And for those who might not be familiar with Antonis's work, he conducts like 3 broad areas of research, and these are some posters that I had made for him. One of which is trying to understand evolutionary relationships between organisms, as well as the parameters that influence our inferences. He also studies the evolution of human pregnancy, as well as the evolution of yeasts and molds. And in this realm is where a lot of the work I've done falls, and more specifically, it falls under the umbrella of trying to understand the rapid tempo of evolution among fungi. So to this end, I've completed or am working towards completing 3 different aims, one of which is studying rapid, ancient evolutionary events, more recent rapid evolutionary events, as well as developing methods and tools that can help us study these processes. So when it comes to an ancient form of rapid evolution, I've studied this hypermutating yeast lineage that has lost a whole bunch of genes that were very unexpected to have observed to be lost, such as DNA repair genes and cell cycle genes, and those seem to be associated with a faster evolutionary mutational rate in these organisms. When it comes to the software, I've developed a handful of toolkits. Some of them are very, very simple, such as OrthoFisher, which helps extract genes using HMMs, as well as ggpubfigs, which helps you create publication quality figures in R, but importantly, using colorblind friendly color palettes. So it helps you make your science accessible to more people. Some of the more complex software I've developed include ClipKit as well as PhyKit. And PhyKit is a broad toolkit for processing and analyzing multiple sequence alignments as well as trees. And then ClipKit is this alignment trimming algorithm, this multiple sequence alignment trimming algorithm. And so just to go on a brief escapade about ClipKit, this has been a really fun project and one of my first endeavors into software development. And this was primarily motivated by a 2015 study that had reported that most multiple sequence alignment trimmers actually worsened your single gene phylogenetic inferences. And their take-home message was that the more aggressive the trimmer, the worse it performed. So they didn't explicitly say this, but this suggests that current methods are actually accidentally removing sites with phylogenetic signal, sites with phylogenetic certainty in them. So motivated by that, I felt like there was this need to develop an alternative alignment trimming strategy. And so instead of following the pattern that most multiple sequence alignments follow, something that was proposed in the early 1990s, is rather than removing sites with uncertain phylogenetic signal, what if we just focused on those that we knew had phylogenetic certainty or phylogenetic signal, something that is less gray in its definition? And so that's really the basis of ClipKit. That's what ClipKit does. And without going into even more details, I'll just show you how it performed compared to other alignment trimmers. So your y-axis is best performance on the bottom because it's a rank based. So the lower the rank, being in first place, the better it performs. And then as you go higher, it performs worse. On the x-axis, they're sorted from best performance on the left and worst performance on the right. So this particular data set is with simulated data. I've also done the same sort of tests for empirical data. But if you're wondering why no trimming is performing so well, it's in part because it's simulated data. No trimming also just seems to perform well, as reported in that 2015 study. So this ranking is the summary of examining tree topology accuracy, tree support, and then we also looked at the accuracy of branch length estimation. So ClipKit has all the blue stars above it, and you can see that performed really, really well across the various modes and like nuanced styles of trimming that ClipKit offers. So I'm really happy to see that ClipKit is out, and I welcome people to use it, and I also welcome feedback about its usability. When it comes to rapid evolution for more recent events, this is what I'll spend the rest of my time talking about. And it has to do with hybrids in fungal pathogens. Before getting into fungi, I just want to back it up and talk a little bit about hybrids that we're all familiar with. And I think the one that everyone knows is the mule. So the mule, it's so iconic because it's been used to bridge physically distant cultures and acquire physically distant resources. And this is in part because of a phenomenon called hybrid vigor, wherein the mule gets like the best traits from both of its parents. So from the donkey, it has strength, patience, endurance. From the horse, it has athletic ability and speed. And the only reason this hybrid is possible is that despite the donkey and the horse looking a bit different from one another, they're actually really similar to one another, genetically speaking. They're less than 1% diverged. And so this type of a hybridization event could be classified as a hybrid speciation because it's kind of a new species being born out of two distinct species hybridizing together, which is distinct from introgressive hybridization. Again, the mule, though, is an evolutionary dead end, so it's sterile. That isn't always the case for fungal hybrids. So there's been a lot of work for fungal plant pathogens as well as yeast human pathogens that have hybridized, and this has contributed to hybrid speciation. And then there's been a lot of examples of introgressive hybridizations, and those are a bit easier to find because they leave behind this phylogenetic footprint. So today we'll be talking about hybrid speciation, wherein two different filamentous fungi came together and created a new species. So that's where the title comes in. When two become one, the hybrid origin of a filamentous fungal pathogen. So I'll talk a little bit about Aspergillus fungi, then talk a little bit about the genomics of the hybrids, and then the phenotyping of the hybrids. So Aspergillus, it's a genus that is home to many deadly fungal pathogens. They pose a clinical, economic, and social threat to humans. They're responsible for 200,000 plus infections a year. However, when you look across the tree of Aspergillus fungi, not every single Aspergillus species is a pathogen. So animal pathogens have been labeled in a maroon circle, and this suggests that there are evolutionary forces that might lead to certain organisms being able to cause disease in animals, and thus, driving the evolution of pathogenicity. So we wanted to understand what types of evolutionary forces lead to pathogenic organisms, and we decided to take a focused investigation on just one species, and that is Aspergillus nigilans. It primarily infects those with chronic granulomatous disease, commonly referred to as CGD, but it's been related to other lung-related diseases. And so what we did is we acquired Aspergillus nigilans from the hospitals, we grew them up on plates, we did genome sequencing, and we were also able to do phenotyping. Now, we knew that they were Aspergillus nigilans because that's what the clinicians had told us they were treating it as, and that's what they had identified the species as. And in the clinic, this has to be done fast. And sometimes this is done using MALDI-TOF, mass spec, or sometimes just with microscopy. So you could imagine how that might lead to some misidentifications. And that's exactly what we saw here because these genomes did not look like any Aspergillus genome that we had seen previously. A typical Aspergillus genome has around 10,000 genes, a genome size of 30 megabases, and on the z-axis, very few duplicated BUSCO genes or these otherwise universally single copy genes. But these genomes, these clinical isolates shown in purple, they had twice of everything. They had twice the genome size, twice the number of genes, and many of their genes were present in duplicate. So we conducted molecular phylogenetic analysis to try and determine what exactly had we sequenced. And what we found is that they had two loci for all of these very taxonomically informative loci that are usually single copies. So calmodulin, Bene, and each locus fell into distinct clades of the Aspergillus tree. One which is blue and is it monophyletic with Aspergillus spinulosporus, and then one that's in red and it's sister to Aspergillus quadrilineatus. And so just to show you one isolate and its two loci falling into two different clades, this is clinical isolate ASFU 2033, and you can see how it has this one blue copy and this one red copy. So through additional genome sequencing, additional phylogenetics, comparative genomics, we were able to pinpoint the hybrids, the parents of the hybrids to Aspergillus spinulosporus, and a close relative that we don't know yet, this Aspergillus quadrilineatus-like species. And these two came together, they hybridized, and they formed Aspergillus latus. Now we propose a model by which this happened, but we're not entirely sure. But we think that Aspergillus spinulosporus and this quadrilineatus-like species must have been growing close to one another and then undergone cellular fusion, then forming this heterokaryotic stage, following nuclear fusion, and then forming Aspergillus latus, this diploid allopolyploid. Interestingly, it's not an evolutionary dead end like hybridization is for the mule. The asexual spore, it can form both asexual spores and sexual spores. So now that we had the genomes of this Aspergillus latus hybrid, we wanted to next try to determine what are the features of its genome and how stable are each of its parents within the genome. So what we did is we assigned each gene to a parent of origin. And the way that we did that was to calculate sequence divergence of each gene to the known parent. And then we just plot out their histogram. So we'll have y-axis being gene count and then the x-axis being sequence divergence. And what will fall out in this type of analysis is that you'll get a bimodal distribution where one distribution are genes that have very low divergence. So those are all genes from that one parent. But then there'll be a second distribution, and these are all genes that are substantially diverged, and they're from the other parent. And you can look at the proportion to which they both occur in the genome using pie charts or whatever else you may like. So if we think about the mule, this is like we were able to get all the parents from one parent, all the genes from one parent, and then partition out all of the genes that came from the other parent. So this is the type of distribution that you would expect for a hybrid. And this is the type of distribution that we saw across all of the hybrids that we had identified where they had this bimodal distribution that allowed us to assign each gene to a parent. And both parents were pretty much equally represented in every hybrid genome. And just to highlight that same strain that I showcased during the molecular phylogenetics, here's this ASFU 2033 with that bimodal distribution. So now that we had created maps of all of these hybrid genomes, we conducted analysis of gene pairs, we looked at recombination, we looked at copy number variants and aneuploidies, and all of that analysis revealed that these hybrids are actually very genetically stable, which is distinct from what typically happens in a hybrid genome, which is rapid decay or sometimes rapidly going to like a single copy state, especially for certain types of genes. So with that, we have this new hybrid organism. And one other question that we wanted to address is how different are the parents? And what we found is that they're about as different as a human is to a lemur. So roughly 8% diverged, which is pretty substantial compared to the mule. But when you look at other fungal hybrids, it's pretty much on par with what you might expect. So next, I'll go into the phenotyping. What we found is that the hybrids exhibited phenotypic variation both at the species level and within species and between species. And what we did is we examined virulence, interactions with human immune cells, drug susceptibility, growth in various conditions. And to summarize all of the phenotyping, we can present that information in a PCA plot. So the hybrids are in purple, and you can see they're pretty diffuse along the x-axis. But they're actually very similar to one of their parents, at least, Aspergillus spinulosporus, the parent that we're sure of. But they're quite different from Aspergillus quadrilineatus, and that might be because Aspergillus quadrilineatus isn't the exact parent. But one thing to note is that they're very different from Aspergillus nigilans, which is shown in the bottom left of the principal component analysis. And this is what they were originally identified as in the clinic. Just to show an example of one trait that exhibited phenotypic variation within Aspergillus latus, within the hybrids, is the Galeria kill curves, or virulence, a measure of virulence. And so in this kill curve, what we found is that sometimes Aspergillus latus was the most virulent strain, or that we had examined, ASFU 1710, because it killed Galeria, this invertebrate moth, the fastest. But sometimes Aspergillus latus was also the least virulent, such as that Mo 46149 isolate that did not kill very many Galeria or very many moths. So this demonstrated a lot of strain heterogeneity within Aspergillus latus. Even though sometimes we saw a lot of within species heterogeneity, sometimes we found that Aspergillus latus was more similar to one parent compared to the other. So for drug susceptibility, the Aspergillus latus strains were more like their blue parent, Aspergillus spinulosporus, whereas for resistance to hyphal killing by human immune cells, they were more like Aspergillus quadrilineatus, their red parent, so to speak. Interestingly, in both cases, this was the more robust parent. So they were more drug susceptible, or sorry, they were less drug susceptible, and they were more resistant to hyphal killing by human immune cells. And so this is very reminiscent of the mule, where it has some of the best traits from both parents. And so we hypothesize that these might be signatures of hybrid vigor, and this is something we're investigating further. So in summary, this is a current biology paper titled Pathogenic Allodiploid Hybrids of Aspergillus, wherein we sequenced these clinical isolates of Aspergillus fungi. They were originally typed as Aspergillus nigilans, but when we took an in-depth investigation of their genomes, we realized and found evidence for allodiploid hybridization and a surprising amount of genome stability amongst these clinical isolates amongst the hybrids. Next, we did a lot of phenotyping, and we found that the hybrids differed from each other as well as from both of their parents, or sometimes they were more like one parent and less like the other. Now, the reason that I think that this is a contribution that I'm proud of, at least, is because I think it's important for two reasons. Medically, we show that it's really important to have accurate isolate identification in the clinic. And perhaps this will move towards the idea of taxogenomics in the clinic, as well as we also emphasize strain heterogeneity. And then evolutionarily, there's the plant pathogens that are hybrids, there's the yeast pathogens that are hybrids, but to my knowledge, this is the first account of a filamentous fungal pathogen that's a human pathogen and a hybrid. At least that's, I believe that's true, at least for Aspergillus fungi. So with that, I do want to thank the really amazing and a big project team that helped make this project possible, especially the co-first author, Abigail Lind, who's now a Gladstone Institute Fellow at UCSF. And then Gustavo Goldman is a co-senior advisor on this project, and there's so many other people that contributed greatly to making this happen. I also want to thank the ROCUS Lab, as well as my funding from the Howard Hughes Medical Institute via the James H. Gilliam Fellowship. So with that, thank you very much again for listening in. Thank you for the opportunity. I'd be happy to take any questions.
Wow, claps for you, Jacob. Great job.
Thank you.
Do we have any questions for him? And you're welcome to unmute yourself to speak. Oh, Miguel, go ahead.
Yeah, for me, it's surprising this genomic stability, because I'm quite familiar with the GEs, and they do really weird stuff. So, do you think that all these Aspergillus latus come from the same hybridization event, or do you think that this is something that is reoccurringly, like maybe patients that have one Aspergillus infection, they tend to inhale the other one and they end up hybridizing or something like that?
Yeah, no, I think that's a great question. So in that study, we had 7 isolates that were hybrids. So from just 7 isolates, we didn't really feel comfortable making a strong statement about how many hybridization events have there been. And neither did we feel comfortable talking about how long ago the hybridization was. So perhaps the genomes are fully present and they appear stable because the hybridization event happened like a year ago, or a year ago in evolutionary time. So I think those are some of the things that could contribute to the appearance of genome stability. But yeah, we were surprised about that too.
Awesome, thank you. Any other questions?
I have a very quick question. So what does this mean, for treatment for people, that have Aspergillus, and like, would treatment be the same or would it be different?
Yeah, often treatment is dictated by like a phenotypic profile. So if you're going to administer some drug, you need to know the rough MIC, or minimum inhibitory concentration, of that drug. And clinicians have this like lookup table where there's 1 MIC per drug per species. So some of the things that we tried to emphasize is that different strains had different MIC levels. So there's that strain heterogeneity component, as well as the sort of like know thy enemy approach to genome sequencing in the clinic. So if genome sequencing occurs in the clinic, you'll know for sure what you're dealing with, as opposed to just like taking a quick glance. I know, the access to different materials is different across the world. So it's definitely a hurdle that hopefully will be overcome at some point. But I think in the clinic, what this really suggests is, in the future, in the most ideal setting, we would have this taxogenomic approach where we can sequence genomes right then and there, and then sort of tailor disease management strategies to the genome characteristics that we identify.
Thank you.
Awesome, thank you. Any more? Okay.
All right, thank you very much, everyone.
Awesome, thanks so much. All right, so we're gonna go ahead and move on to our next speaker. So the next speaker is Dr. Abigail Labella. So she's a postdoctoral scholar in the same lab, Antonis Rokas' lab in Vanderbilt University. Her research career, she has studied many different organisms, including beetles, humans, fungi, lizards, and deep sea mollusks. Her graduate work in the lab of Clifford Cunningham at Duke University took her all the way to the bottom of the ocean in regards to research using the submarine Alvin. This breadth of work is tied together by her interest in harnessing genomics to answer questions about the world around us. While her current research projects include codon usage in fungi and the evolution of human pregnancy, she currently shares her home in Nashville with her partner, 2 dogs, and 2 cats. Ab, you're welcome to go ahead.
Thank you. I'm going to get sharing my screen. Get a little highlighter. or laser pointer. Okay. Thank you so much for having me. Today, I'm going to talk a little bit about my work that's harnessing synonymous codon for reverse ecology in budding yeasts. So like you heard, I have 4 potential co-presenters that might make an appearance. You may have seen the smallest one, but I apologize if they interrupt. They're usually fairly well behaved, so you might see them. But as you heard, I've had quite a long career in research. This is actually a picture of me in high school working in a lab on aberrant mRNA transcripts, working with yeast. And I swore after my summers working there that I would never work with fungi again. So that was not true. I worked for a summer at the Woods Hole Oceanographic Institute as a budding marine biologist, studying how killifish adapt to estrogen-like compounds in their environments. Then during my undergrad research, I studied allometry in beetles, or how they determine different body sizes. As you know, beetle can have some extreme morphologies when it comes to body size. Then after graduate school, I spent a year working at the Children's National Medical Center, studying a very rare urea cycle disorder that's determined genetically and looking at potential treatments for that. And then, like you heard, during my graduate work, I spent my time studying population genetics of deep sea mollusks. This is me about to climb into the submarine Alvin to go sample my organisms at the bottom of the ocean. So even though I've sort of gone across the tree of life, what's bound together my interest has really been genetics. I'm really interested in knowing how genes determine different things about organisms. And so you can see that this has been applicable across a wide range of species. And so what I've done now in my postdoc work, and what really ties together my work in pregnancy and in genetics, is looking at whole genomes and really looking at a much broader scale for genomes. So what can whole genomes tell us? What kind of information can we get when we're not really limited by looking at individual parts of a genome? And this is a very popular genome right now. This is the genome of the SARS-CoV-2 virus. And so you can see that even very small genomes can cause a lot of havoc. So like you've heard, I'm a part of the ROCUS lab at Vanderbilt here in Nashville, Tennessee. We obviously now meet virtually, but it's a vibrant lab with lots of different components. But the work I'm going to talk about today is my work in fungi. So here's another one of Jacob's great artwork. The budding yeasts are probably the least charismatic of the fungi. I'm not going to be talking about any of the really ones with cool morphologies, but Jacob has done a great job showing that there is a lot of diversity in the budding yeast. They are fantastic. So for those of you that aren't as familiar with the budding yeast, I'm going to give a little bit of background about who they are and why I think they're really cool. Obviously, they're a subphylum of fungi known as the Saccharomycotina. It contains Saccharomyces cerevisiae, probably the most famous member and the one we like to enjoy in our own homes. But what you might not know is there's a little more than 1,000 known species within this group that live on incredibly diverse ecologies. So basically anywhere we look on planet Earth, we're likely to find some fungi, anywhere from fecal matter to our own bodies, to oceans, to biotechnology, there's going to be fungi there in the form of budding yeasts. They also have a huge diversity of metabolic and growth phenotypes. So one of the reasons that they can establish themselves in such diverse ecologies is that they can metabolize a huge range of metabolites. Things like glucose and ethylamine and sucrose and trehalose. Basically, if it's a carbon compound, there's likely to be a fungi that can take advantage of it. They can also grow on a wide variety of temperatures, so ranging anywhere from below freezing, or not below freezing, but getting close to freezing up to higher temperatures, including our own body temperatures. And this is really facilitated by a large genetic diversity. So the difference between two species within the Saccharomycotina, Saccharomyces, and one of the most distantly related, Yersinia lipolytica, is more than one amino acid substitution per site. And just to put that into context, this was work that Jacob did, where you can see that that's about the difference between humans and roundworms or Arabidopsis and green algae. So we're really talking about a hugely diverse group when we're talking about genetics. This really makes them a model clade for evolutionary studies. We have huge diversity of metabolic and growth phenotypes, a huge genetic diversity, a huge ecological diversity. We can start to ask some really interesting questions about evolution. And this is where the Y1000 Plus project came into being. The aim of this project is to phenotype and sequence the 1,000+ Saccharomycotina species. And this is really a global effort of researchers from Vanderbilt, University of Wisconsin, Villanova, China, and the Netherlands, a large group with a lot of expertise that are working together to complete this aim. And we are approaching that 1,000 mark soon. So keep an eye out for that. But my work and what I'm really interested in is the idea of being able to connect phenotype to genotype to environment. So how can we look at genomes and understand something about phenotype and environment by looking at genomes alone? And so trying to complete this circle of phenotype, genotype, and environment by looking at the information we have in the 332 genomes. And you may be wondering why I have a codon table under genotype instead of another genome. If you follow me on Twitter, you probably know why. And that's because I am absolutely obsessed with codons and I find them totally fantastic. So codon usage, as you probably know, is the usage of different synonymous codons to encode the same amino acid. So for almost all of our amino acids, there's more than one choice of which codon to use. And despite what you may have remembered from biology class, synonymous codon usage matters. If there's only one thing you remember from this whole talk, I want you to come away with knowing that synonymous codon usage is not silent. And if you need more proof than that, which I'm going to give you today, you can follow me on Twitter because every week I feature a new story on Third Base Thursday of why codon usage matters and how we can use it in ecology and biotechnology and disease treatment. So this isn't just in fungi, this is across all organisms. I highlight the latest research going on in codon usage. But simply, and for the purposes of this talk, I'm going to talk about how codon usage actually can affect translation via the tRNA pool. And so this is basic biology, but I promise I'm going to move through it quickly. Here I have my little budding yeast cell. And within the nucleus, we have our tRNA genes on the chromosomes. And the tRNA genes are found in different copy numbers. So sometimes you'll get 4 or 5 copies of a tRNA gene, and other tRNA genes have just 1 copy. And so when you look within the cell, the abundance of these different tRNAs is roughly corresponding to the number of genes within the tRNA. So we use that as a proxy. So you're wondering, why am I talking about tRNAs? Well, codon bias actually influences the accuracy and speed of translation elongation. So I'm going to use an example here of our little mRNA molecule. We've got 2 different synonymous codons in green and in blue. They encode the same amino acid, so this would just be like glutamine, 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 glutamine over and over again. We have in green a globally frequent codon that is decoded by this abundant tRNA, and in blue we have this globally rare codon that's decoded by this sparse tRNA. And when we have enough of this green tRNA to go around, this ribosome can zip along and make plenty of this protein. The opposite scenario, where now in the same cell, we have this transcript with a lot of this globally rare codon. What happens is the ribosomes actually can't find enough of that tRNA. They can't be recharged fast enough, and we actually can get stalling on the ribosome or much slower protein production. And that's where we get these 2 different scenarios. We have codon usage that matches the tRNA pool, and this creates fast and efficient translation, high protein output, and stable mRNA. And codon usage that does not match the tRNA pool. This is slow and inefficient translation, low protein output, and targeted mRNA decay. And we call this top 1 codon optimization, or optimized codon usage. So why does this matter? Why do we care about whether or not optimal codons are used? It's because there's a selective pressure for fast and accurate translation in highly expressed genes. So there's a relationship between being highly expressed as a gene and having optimal codon usage. And this is a fact that we like to take advantage of because we can't always do expression studies in the lab. And so when we look at expression data, we have high expression up here on the y-axis and codon optimization on the x-axis. We see that there's a fairly strong positive correlation between codon optimization and high expression, which means that if there's a selective pressure for codon optimization, we can use it as a proxy for gene expression without doing any expression work in the lab. So we needed to know first, in our budding yeast, is there selection on codon usage? Can we use codon usage as a proxy for expression? So we started out with a subset of 332 genomes. We then assessed the tRNA genes within the genomes and then assessed the codon usage in the coding sequences and used this program called the species-specific tRNA adaptation index to tell us how optimized all of these coding sequences are. Then we do this thing called the S-test, where we ask, is that selective pressure on the codons associated with the codon adaptation index? Basically, this is just a fancy way of saying if these two things are related, positively correlated, we think that there's selection acting on codon usage, and this species is a good candidate to use codon usage for expression. And then this one would say there's no relationship, and there's probably mostly genetic drift responsible for codon usage. And in the 332 species, we actually find examples of both of these. That species I showed before, where we had empirical evidence that codon usage is associated with expression, we see this positive correlation. And in another species here, we don't see that correlation, suggesting that drift is probably responsible for codon usage. So all that is to say, for the vast majority of the 332 budding yeast species we looked at, we do think there's translational selection on codon usage, and therefore we can use codon usage as a proxy for gene expression. So what is our hypothesis? Well, we wanted to know if we could detect an evolutionary signal for adaptation in high expression in particular genes. And so this was our hypothesis, that we would have some ancestral species that has gene Z. And gene Z encodes enzyme Z that turns metabolite A into metabolite B that can then be used in carbon metabolism. So this species then gets split into two populations, one in which we see a lot of compound A and one in which we don't see a lot of compound A. And because now there's a selective pressure to increase the expression of this gene Z, we're going to ultimately see a high codon optimization in Z that's associated with this environment versus no codon optimization in gene Z in an environment where that compound is not present. So this was our hypothesis. It was great, but where do we start? We had to pick a place to test to see if we can actually detect this signal. And so we decided to start with the galactose pathway. So galactose metabolism, or the Loire pathway, is a famous metabolism pathway in budding yeasts and humans. It's used all of the time in biotechnology, and it's composed of three enzymes, GAL1, GAL10, and GAL7, that converts galactose into glucose 1-phosphate that can then be used in carbon metabolism. So the first step was to, in all 332 genomes we were looking at, here's a phylogeny, and they're just colored by the different subgroups. And so what we were looking at is to see if we could find those GAL genes in here. And anywhere where you see a bar, that means we were able to identify GAL1, GAL10, or GAL7. And the color of the bar, so the darker the color, means the more highly optimized that gene is within the genome. And you can see that there are some clusters where all of the genes are present, but they're not very highly optimized. And then you can see some clusters where we have all of the genes are highly optimized in this pathway. So we would predict that gene expression of the GAL pathway in these species with high codon optimization is much higher. So, well, can we test this? How do we know if this is true? Well, one of the things that we also had in hand were growth phenotypes from Dana Opulente. And so she took growth on galactose. So we had a growth rate associated with galactose. And we looked to see, is there increased growth with optimal codon usage in the GAL pathway? And what I'm going to be showing you is a phylogenetically independent contrast, which means that this is corrected to account for the fact that all of our species are related. So we know that these are not independent observations. They're in fact related to each other, and this regression takes that into account. And so what we see is for GAL7, codon optimization is highly associated with growth rate on galactose. And so we are now thinking we can use this as a proxy for growth rate on galactose and expression. This is true in GAL1 and GAL10 as well, although it's not as significant in GAL1. And there are some biochemical reasons we think GAL7 might be the most highly correlated, including the fact that the compound that is metabolized by GAL7 is a toxic intermediate. So if you don't have enough GAL7, there can be the accumulation of that toxic intermediate. So this might be the most important key or cog in that metabolite. So we did this, and we also wanted to know when we have new species, can we actually predict growth rate from codon usage? So this is another representation of the association between codon optimization on the x-axis and growth rate on the y-axis. We had 2 new species. So we said, okay, based on their codon optimization, can we predict, is this a good prediction for what their actual growth rate might be? And we found that it was. So this was the actual measured amount of growth rate on galactose. And so we're doing a fairly good job of being able to predict growth from the optimization of a single gene, which is pretty fantastic. So I said I was going to talk about environment. We've talked about the relationship between genome and phenotype. And now I'm going to talk about phenotype and environment connected to genotype. So the Saccharomycetaceae, this is a subgroup of the Saccharomycotina. This contains our Saccharomyces cerevisiae and other commonly found yeast that live on alcohol substrates, dairy substrates, or some of them are found on both. So we divided our species into these categories and asked, do we see any differences in codon optimization? And what we found was that in species found only on dairy, they had a much higher optimization in GAL1, GAL10, and GAL7. So this suggests that high growth rate on galactose is associated with dairy. Now that probably makes sense, as we know that cow's milk is made up of lactose, and there's a whole microbiome associated with it that converts lactose into glucose and galactose. So in order to be able to take advantage of galactose in the environment, which can be up to 5%, there's an advantage to growing quickly and utilizing the galactose within those environments. So we made a second discovery in this CUG SIR1 clade, and in this case, we divided the organisms, the species, into those that are found associated with insects, those that are found associated with humans and or insects, or neither of those 2 categories. You might be wondering, why did we include humans or humans and insects? It's because of all of the species we found that are associated with humans, so being found on the skin or in infections, they're almost all associated with insects. There were only 2 that we found that had never been isolated from an insect before. And so this CUG SIR1 clade is actually that Candida albicans group that you might have heard of that can colonize human skin. And so we look to see if there's different optimizations associated with these ecological groups. And it's less significant, but we do see that there is an association with being associated with humans and an increased expression or inferred expression through codon optimization in these species. So why would that make sense? You know, we're thinking, is there more galactose in that environment? Why would especially GAL7 and GAL10 be associated with the ability to grow on humans? So there are some hypotheses that we came up from the literature. One is that there's low glucose availability in the digestive tract. We like that glucose, we're gonna utilize it. And so a group found that in Candida albicans, during colonization of human host tissues, there was upregulation in GAL10. But it also might not have to do with carbon metabolism. So there's some suggestions that GAL10 is involved in cell wall formation. So cell walls can protect these species from the human host. And so a group saw that they had decreased survival in hydrogen peroxide when you delete GAL10. So there may be something more than carbon metabolism that we've stumbled upon by using codon optimization. So I hope I've convinced you that I've been able to connect a specific genotype, so codon optimization in the GAL genes, with specific phenotypes and environments. So we think that this is a good base for which we can then move on to the 1,000 genomes. So this is a preliminary tree associated with the 1,000 genomes. And so with this, we're going to use a phenotype first analysis. And this is really made possible by pioneering these taxonomists, including the late Cletus Kurtzman, who was one of the founding members of the Y1000+ project. And these groups have meticulously characterized the phenotypes of these yeasts, including things like growth at 37 degrees, which is associated with growth on humans. So 37 degrees was chosen because it is associated with growth on humans. And so what we're looking to do now is taking the whole global genome and looking at all of the genes that have high codon optimization, and therefore we can infer are highly expressed. Within this group, a lot of these are ribosomal genes, so things that we know are highly expressed globally and constantly. And so we're looking to see if any of these highly expressed genes we can differentiate in those species that can or cannot grow at 37 degrees. So we have a subset of species that can't grow and a subset that can. Then we'll pull out which genes we infer to be the most highly expressed and see if there are any genes that are specifically highly expressed in those species that can grow at high temperatures. So this is what we're doing next once we get all of the 1,000 genomes in hand to see if we can come up with new genes that are associated with growth phenotypes, and then we're going to test them in the lab. So you're convinced, you're so into codon usage, you're gonna go into your own organisms and start testing things. I just wanna talk a little bit about the advantages and disadvantages of this method. So one of the cool things is that I'm able to predict growth from codon usage in a single gene. I'm only using genomic data, and we really think this can be a powerful tool for species that are difficult to grow in the lab, that are difficult to work with, or are really dangerous to work with. There's the opportunity to study growth or study phenotypes inferred from codon usage. So we're also inferring selection on high expression, and this is really a nuanced point, but we're not actually looking at what's happening in a lab. So we can do expression work in the lab, but that's really only going to tell you something about expression in the lab. And when we're looking at codon optimization, this is actually the signature of an evolutionary pressure for high expression. So we're really inferring expression that has been high over the evolutionary history of this organism. And that may provide additional insights for pathways that we can't get expressed. So there are species that have all 3 gal genes, they have them at a decent codon optimization, but we can't turn them on in the lab. So is that because they've lost the ability to do that? Or is that because we're not giving them the right signals? There can be information in that. And we're also able to look at every gene in the genome. Obviously, the better our gene predictions are, the better the output's going to be, but we can really look at all of the genes, whether or not they're expressed in the lab. And the disadvantages are that, codon usage is not under high selection in all species. There's some evidence that we had in our first paper that even if it's globally not under selection, that there could be a subset of genes within each genome that have this selective pressure. And one of the things that I'm really interested in, but is also a disadvantage, is that the identity of optimal codon usage is really tied to the dynamics of the cell. So whether or not tRNAs are expressed at high or low levels. And we know that this changes throughout things like the cell cycle or in response to stress. So there can be a completely different optimal codon landscape in different moments of the cell's life. And that's something that we need to investigate further because we really don't have a good understanding of how this dynamic changes over time or between environments. So I want to acknowledge everyone that's worked on this, especially in the Rokas lab, Jacob Steenwyck and Marie Claire, people in the Hittinger lab, the other Y1000 members, Dana Opulente, who is the growth and phenotype czar and genome producing czar. She's been amazing at all of this. Our funding sources, all of the creative people on the internet who I have taken their images from and given them partial credit here. And I've got time for some questions, and feel free to tweet at me, email me, or submit something on my website if you want to fill out that form there. But email or Twitter is probably the best place to get in touch with me. So I'm happy to take any questions now.
Wow, awesome work, Abby. So earlier Miguel had a little, statement he posted up on the chat. So he mentioned that maybe human-associated yeasts establish themselves early on when diet includes lots of milk.
That could definitely be a part of it. I don't think we have a solid idea of why galactose metabolism is associated with growth phenotypes. So it'd be interesting to do some experimental work to validate can upregulating these gal genes increase colonization in the lab? So our reviewers on that paper have been begging for more experimental work, but it's not in the cards for right now, but definitely things we want to do in the future.
Yeah. Miguel, did you have any follow-up suggestions or questions about that?
No, that was just an idea that I decided to pop up in the moment. I thought it could be, a hypothesis.
That's great. Yeah. Cool. Any other questions?
mean, I'm a bit intrigued to know if you have tried this codon optimization thing in outside the Saccharomycotina, in other fungi or in. I think I've read somewhere that human genes are also codon optimized. So it may be something quite universal, but I'm worried that the speed at which genes typically grow may be optimized in everything, like codon termination in bacteria and things like that.
Yeah, so most people have found codon optimization or selection on codon usage in microbes. That's where it's gotten the most traction for sure. Neurospora is where a lot of the ongoing work for characterizing codon usage across circadian rhythm and during the cell cycle has been done. So that's become a great model organism for codon usage. For a while, it was thought that in humans this doesn't exist, but emerging evidence is looking like it might be tissue specific. So one of the things that I'm really intrigued by is the thought that by altering one or two tRNA species, you can actually globally regulate a whole set of genes that have a similar codon usage. So if you have a bunch of genes that use a really rare codon, if you have a cell type or a tissue type that expresses that tRNA highly, in that subtype of tissues, you might get optimization that's specific. So there is also some good work coming out of Drosophila that looks like it may actually be really tissue-specific codon optimization to help regulate a set of genes. And there's some also really cool things about maternal mRNAs being regulated, differentially regulated based on codon optimization.
Awesome. Thank you.
Thank you very much. Excellent talk.
I'm obsessed with codon usage, so I'm always reading about it.
That's great. I mean, I have like a very, very basal knowledge of this. I mean, I work with yeast, but I work with a black yeast. And I'm sure there are codon differences there too, but how would one person start to look into that?
Yeah, so the relative synonymous codon usage is a fairly standard thing that you can do in a program called Codon W. And basically you just need gene annotations and you can look for differential codon usage between species. So a project that Jacob and I were working on for a little while was looking to see if we can identify horizontally transferred genes based on how codon usage changes. Some of the evidence from actually the Gal pathway, horizontal gene transfers in the Gal pathway looks like it hasn't been differential codon usage hasn't been a big obstacle for some of the horizontally transferred genes within our group. But there's some basic things, as long as you have gene annotations, Codon W will spit out some good data for you.
Awesome. Okay. Thank you so much for that. I'll definitely look into it.
And you can always follow up with me and I can point you to other directions if you're interested.
Cool. Yeah, we'll definitely do that. Any other questions? All
awesome.
right.
Thank you, everyone.
Yes, thank you so much. So as we do every month, we're planning on having more speakers come in, 2 speakers presenting their work, hopefully try to represent different labs. And so I appreciate having 2 members of the same lab represented here. You're welcome to contact us. We have our Twitter, we have our social media accounts, but we also have email. So you're welcome to reach out and say that, hey, I'm interested, or I have a lab mate who's interested, and we'd love to feature you. Okay. Awesome. Well, thanks so much, and see you next time. Okay. Bye.